Welcome to a journey into the world of lichens, those remarkable organisms that cover rocks, drape from trees, and thrive in places where almost nothing else can survive. Tonight, we'll explore the quiet partnerships that make lichens possible, the strange beauty of their forms, and the extraordinary ways they've adapted to life on Earth. Let's begin with something fundamental. Something that might surprise you if you've never thought carefully about what a lichen actually is. A lichen is not a single organism. It's not a plant, not a fungus, not an alga. It's something more interesting than any of those. A lichen is a partnership, a symbiosis between a fungus and one or more photosynthetic partners. These partners might be algae, or they might be cyanobacteria, or sometimes both. 2 or even 3 different organisms living together so intimately that they create what looks and functions like a single being. It's one of nature's most successful collaborations, a relationship that has allowed these partnerships to colonize nearly every environment on Earth. The fungal partner in this relationship has a name. Scientists call it the mycobiont. Myco comes from the Greek word for fungus, and biont simply means living thing. So the mycobiont is the fungal living thing, the fungal half of the partnership. In most lichens, the fungus is the dominant partner in terms of sheer biomass. It provides the structure, the body that we see when we look at a lichen. The fungus builds the framework that holds everything together and protects the more delicate photosynthetic partners from environmental stress. The photosynthetic partner has its own name too. Scientists call it the photobiont. Photo for light, biont for living thing. The photobiont is the organism that captures sunlight and converts it into chemical energy through photosynthesis. This partner might be an alga or it might be a cyanobacterium, but whatever it is, it's performing the crucial function of making food for the partnership. Without the photobiont, the fungus would have no source of energy. The photobiont is essentially the lichen's solar panel, its power plant, its food factory. When the photobiont is specifically a green alga, there's an even more specific term for it. Scientists might call it a phycobiont. Phyco refers to algae in general. It's a more precise term, telling you not just that there's a photosynthetic partner, but that it's specifically an alga rather than a cyanobacterium. Many lichens use green algae as their photobionts, and these algae are typically unicellular organisms that live inside the protective structure the fungus creates. But when the photosynthetic partner is a cyanobacterium, one of those ancient bacteria that can photosynthesize like plants, it gets yet another name. Scientists call it a cyanobiont. Cyanobacteria are remarkable organisms. They're bacteria, which makes them fundamentally different from algae, which are eukaryotes. Cyanobacteria were among the first organisms on Earth to produce oxygen through photosynthesis, billions of years ago. They transformed Earth's atmosphere and made complex life possible. And today, some of them live in partnership with fungi, creating lichens. Now, let's talk about what each partner contributes to this relationship, because it's a beautiful example of cooperation at the cellular level. The fungus builds most of the lichen body. When you look at a lichen on a rock or hanging from a tree branch, you're mostly looking at fungal tissue. The fungus creates the structure, weaving its thread-like cells called hyphae into dense mats that give the lichen its shape and form. This fungal structure provides protection. It shields the photosynthetic cells from intense sunlight that might damage them. It helps retain moisture during dry periods. It creates a stable environment where the photobiont can function efficiently. The fungus is the architect and the protector, the builder of the home where both partners live. The photobiont, meanwhile, produces simple sugars through photosynthesis and shares them with the fungus. This is the core exchange at the heart of the lichen partnership. The photobiont captures light energy and uses it to convert carbon dioxide and water into glucose and other sugars. These sugars are then made available to the fungus, which cannot photosynthesize on its own. The fungus essentially farms the photobiont, providing protection and structure in exchange for food. It's a transaction that has worked successfully for hundreds of millions of years. If cyanobacteria are present in a partnership, they bring an additional gift beyond photosynthesis. They can fix nitrogen from the atmosphere. Nitrogen fixation is the process of converting atmospheric nitrogen gas, which most organisms cannot use, into ammonia and other nitrogen compounds that living things can incorporate into proteins and other essential molecules. This is an incredibly valuable ability, especially in nutrient-poor environments where nitrogen is scarce. Lichens with cyanobacterial partners can survive and even thrive in places where nitrogen limitation would prevent other organisms from growing. They're self-sufficient, able to make their own food through photosynthesis and acquire their own nitrogen from the air itself. Here's something interesting about the photosynthetic partners in lichens. Many of the algal species that participate in lichen partnerships can also live independently outside the partnership. They're not obligate symbionts, locked forever into the lichen lifestyle. In the right conditions, perhaps in moist soil, or on the surface of rocks or tree bark, or in freshwater, these algae can live freely, photosynthesizing on their own, reproducing on their own, surviving without any fungal partner. The fact that they choose to enter partnerships with fungi suggests that there must be advantages to the lichen lifestyle, benefits that outweigh the costs of sharing the products of photosynthesis. The final fact for this segment puts the importance of lichens into broader context. About 20% of all known fungal species live in lichen symbioses. 20%. That's enormous. Think about the diversity of fungi. There are tens of thousands of described fungal species, from mushrooms to molds to yeasts to microscopic decomposers. And of all these fungi, 1 in 5 has evolved to live in partnership with photosynthetic organisms. This tells us that the lichen lifestyle is not some rare curiosity, some unusual exception to the normal ways that fungi live. It's a major evolutionary strategy, one that fungi have adopted again and again in different lineages, in different environments, at different times in Earth's history. As you settle into rest, picture these partnerships. A fungus and an alga, or a fungus and a cyanobacterium, or sometimes a fungus with both, all living together. Each partner contributing something the other cannot provide on its own. The fungus offering structure and protection. The photobiont offering energy from sunlight. Cyanobacteria, when present, offering nitrogen from the air. Different organisms creating something together that none could create alone. A partnership so successful that it exists in 1/5 of all fungal species, in environments from the equator to the poles, from sea level to mountain peaks. The quiet collaboration that makes lichens possible, happening right now on rocks and trees and soil all around the world. Rest now, and let these images of cooperation settle into your thoughts. Let's explore what the word lichen actually means, because the answer is more interesting and more subtle than you might expect. The word lichen describes a lifestyle, not a single taxonomic group. This is crucial to understanding what lichens are. When biologists classify organisms, they typically group them based on evolutionary relationships. All mammals together because they share a common ancestor. All birds together for the same reason. But lichens don't work that way. The word lichen doesn't refer to a group of closely related organisms that all descended from a single lichen ancestor. Instead, it refers to a way of life, a strategy for survival, a particular kind of partnership between fungi and photosynthetic organisms. It's like calling something a predator or a parasite. You're describing what it does, how it lives, not what taxonomic box it fits into. This becomes clearer when you consider where lichen-forming fungi occur in the fungal tree of life. They occur in multiple fungal lineages. The ability to form lichens has evolved independently many times in different groups of fungi. Most lichen-forming fungi belong to a group called Ascomycota, often called the sac fungi. This is one of the major divisions of the fungal kingdom, containing tens of thousands of species. Some of these are lichenized, meaning they form lichens. Others are not. They might be decomposers, or plant pathogens, or mushroom-forming species. The capacity to partner with photobionts isn't something all Ascomycota share. It's something that has evolved within certain lineages of this group. And beyond Ascomycota, some lichen-forming fungi also occur in Basidiomycota, the group that includes most of the familiar mushrooms. Again, only certain lineages within Basidiomycota have evolved the lichen lifestyle. It's a strategy that has appeared multiple times in fungal evolution, not a single ancient innovation that all lichen fungi inherited from a common ancestor. In some groups of Ascomycota, the lichen lifestyle has become particularly successful. Most described species in these groups are lichenized. Imagine a branch of the fungal family tree where the majority of species have adopted this partnership strategy, where living as a lichen is the norm rather than the exception. These groups have radiated, diversified into many species, many forms, many ecological niches, all while maintaining the fundamental lichen partnership. It speaks to the success of this lifestyle in certain fungal lineages, to how well it works as a way of making a living in the world. Now let's consider the other side of the partnership. If there are many different fungal species forming lichens, how many different photosynthetic partners are involved? The answer is surprisingly small. Around 100 species of algae and cyanobacteria commonly act as photobionts. 100 species partnering with thousands of fungal species. This asymmetry is striking. It suggests that being a successful lichen photobiont requires particular characteristics that not all photosynthetic organisms possess. Perhaps the ability to survive being enclosed in fungal tissue. Perhaps the capacity to share enough photosynthetic products to support the fungus while retaining enough for your own needs. Perhaps tolerance for the particular chemical environment that exists inside a lichen. Whatever the requirements, only about 100 species of algae and cyanobacteria commonly meet them. Among these photobionts, certain genera appear again and again in different lichens. Common algal genera include Turbuxia, Pseudoturbuxia, and Rametia. These names might be unfamiliar unless you study lichens professionally, but they represent the workhorses of the lichen world, the algal partners that enable thousands of lichen species to exist. Turbuxia in particular is found in a huge number of lichen species. It's a green alga, unicellular, capable of photosynthesis, and apparently well suited to life inside fungal tissue. Different species of Trabuxia partner with different fungi, but the genus as a whole is central to lichen biology worldwide. There's another algal partner worth mentioning specifically. The orange alga Trendipolia is a frequent lichen partner, but it has a quality that makes it particularly interesting. It can live freely on tree bark and rocks outside of any lichen partnership. Walk through a humid forest and you might see bright orange patches on tree trunks. That's often Trendipolia, living independently, photosynthesizing on its own, not enclosed in any fungal structure. But this same alga also partners with fungi to form certain types of lichens. It leads a dual life, sometimes independent, sometimes symbiotic. This flexibility suggests that the lichen partnership, at least for some photobionts, is more opportunistic than obligate. The alga can do fine on its own, but it's also willing to enter into partnership when a compatible fungus is available. The success and adaptability of the lichen lifestyle becomes clear when you consider where lichens occur. They occur on every continent, including Antarctica. Every single continent. From tropical rainforests to Arctic tundra, from hot deserts to cold mountain peaks, from coastal zones to continental interiors, lichens have found ways to survive. They're not restricted to any particular climate zone or habitat type. They're genuinely cosmopolitan, genuinely global. Few groups of organisms can make this claim, but lichens can. The partnership between fungus and photobiont has proven successful across nearly the full range of environments that Earth offers. In parts of Antarctica, lichens take on an even more prominent role. They are among the dominant visible life forms. Think about that for a moment. Antarctica, the coldest, driest, windiest continent. A place where almost nothing can survive. Where ice covers most of the land's surface. Where temperatures regularly drop to extremes that would kill almost any organism. And in this harsh environment, lichens dominate. They grow on exposed rocks in ice-free areas. They colonize the surfaces of stones. They survive months of darkness during the Antarctic winter, then resume growth when light returns. They tolerate freezing and thawing, desiccation and rehydration, wind and cold that would destroy most other organisms. In parts of Antarctica, if you want to see complex life, you look at the lichens. They're the visible evidence that life can persist even in Earth's most extreme environments. The scientific study of lichens has its own name. It's called lichenology. People who devote their careers to understanding lichens, their taxonomy, their ecology, their physiology, their chemistry, are lichenologists. It's a specialized field within biology, requiring expertise in both mycology, the study of fungi, and phycology, the study of algae, plus knowledge of symbiosis, ecology, and often chemistry. Lichenologists identify new species, map distributions, study how lichens respond to environmental change, explore the chemicals lichens produce, investigate their roles in ecosystems. It's detailed, patient work, often involving microscopy and chemical analysis, field surveys in remote locations, and careful study of specimens in herbaria where dried lichens are preserved for future research. There's even a field devoted to how humans use and perceive lichens. It's known as ethnolichenology. This is the study of the relationships between people and lichens across different cultures and throughout history. How have people used lichens as food, as medicine, as dyes, as materials? How do different cultures name and classify lichens? What folklore and traditions surround these organisms? Ethnolocanology documents the human dimensions of lichen biology, preserving knowledge that might otherwise be lost as traditional practices change. It's a reminder that lichens aren't just objects of scientific study, they're organisms that have been part of human experience for as long as humans have existed. As you drift towards sleep, picture this diversity. The word lichen describing not a single group of related organisms but a lifestyle that has evolved independently in multiple fungal lineages. Thousands of fungal species partnering with about 100 species of algae and cyanobacteria. Particular algal genera like Trabuxia appearing in countless different lichens. Orange Trentipolia living sometimes freely, sometimes in partnership. Lichens occurring on every continent, dominating even in Antarctica's harsh landscape. Lichenologists studying them around the world, ethnolocanologists documenting their roles in human cultures. A lifestyle so successful it has spread across the planet, adapting to nearly every environment Earth offers. Rest now, and let these images settle gently in your mind. Let's look closely now at the structure of a lichen, the physical architecture that makes this partnership work. The body of a lichen is called a thallus. This term comes from Greek, originally meaning a young shoot or twig, but in biology it's used to describe the body of an organism that isn't differentiated into true roots, stems, and leaves. A lichen thallus is the entire visible structure, what you see when you look at a lichen on a rock or tree. It might be a thin crust, or a leafy lobe, or a branching shrubby form. Whatever its shape, it's the thallus, the body that the fungal and photosynthetic partners have constructed together. This thallus is largely made of densely woven fungal filaments called hyphae. Hyphae are the basic structural units of fungi. Each hypha is essentially a tube, a long cylindrical cell or chain of cells, growing at the tip and branching to create complex networks. In a lichen, these hyphae weave together into dense mats, creating the bulk of the lichen structure. The hyphae interlock and press against each other, forming layers and patterns that give the lichen its particular form. Most of what you see when you look at a lichen is this woven mass of fungal hyphae. And within this fungal structure, embedded in a woven network of hyphae, are the photobiont cells. These hyphae surround the photobiont cells, enclosing them, protecting them, positioning them where they can receive adequate light for photosynthesis while being shielded from excess intensity that might damage them. The fungal hyphae are like scaffolding and walls, creating chambers and spaces where the photobiont cells live and work. The arrangement is intimate, fungal cells and photobiont cells in close proximity, separated by just thin cell walls, allowing efficient exchange of nutrients and waste products. Many lichens have layered structures that you can see if you cut through the thallus and examine it under a microscope. There's often an upper cortex, a photobiont layer, a medulla, and sometimes a lower cortex. Each layer has its function, its role in the overall architecture. It's a sophisticated structure, not random but organized, optimized for the needs of the partnership. The upper cortex is the outermost layer on the top surface of the lichen. It's made of tightly packed fungal hyphae, often with thickened walls, forming a protective barrier. The upper cortex protects against intense light and drying. Too much light can damage photosynthetic cells, breaking down the delicate molecular machinery of photosynthesis. The cortex filters light, reducing intensity while still allowing enough through to support photosynthesis in the layer below. The cortex also helps retain moisture by reducing evaporation from the lichen's interior. It's the lichen's armor and sunshade, its first line of defense against environmental stress. Beneath the upper cortex, in many lichens, is the photobiont layer. This is where the algal or cyanobacterial cells are concentrated. They're positioned just below the protective cortex, receiving filtered light, surrounded by fungal hyphae that support them and exchange materials with them. This is the lichen's solar panel, the zone where light energy is captured and converted into chemical energy. Below the photobiont layer is the medulla. This is typically the thickest layer, made of loosely woven fungal hyphae with air spaces between them. The medulla is like the lichen's storage warehouse and structural core. It can hold water in the spaces between hyphae, providing a reservoir that the lichen can draw on during dry periods. It might store food reserves. It gives the lichen structural strength while remaining light and flexible. Some lichens also have a lower cortex on their bottom surface, similar to the upper cortex but on the underside of the thallus. This provides additional protection and helps control moisture loss from below. Other lichens lack a lower cortex, leaving the medulla exposed on the bottom surface. Here's something fundamental about lichen structure. Lichens lack true roots, stems, and leaves. They're not plants, despite sometimes looking plant-like. They have no vascular tissue to transport water and nutrients. No root system penetrating into soil to absorb minerals and water. No leaves with specialized structures for gas exchange. Everything a lichen needs, it obtains directly from the air and rain. It's a simpler body plan than you find in vascular plants, but it's effective for the lichen's way of life. To attach to surfaces, lichens use different strategies. Some use structures called rhizines. These are root-like structures, but they're not true roots because they don't absorb nutrients. They're simply anchors, thread-like or cord-like structures that grip the substrate and hold the lichen in place. Other lichens attach using a central holdfast, a single point of attachment from which the rest of the thallus spreads. Some crustose lichens, those that form thin crusts on surfaces, are so tightly bonded to their substrate that they have no distinct attachment structures. They're simply fused to the rock or bark, impossible to remove without destroying either the lichen or the surface it's attached to. Some lichen species contain something unusual in their structure: crystals of secondary compounds between their cells. These crystals are chemical substances that the fungus produces, often unique to lichens, that accumulate in the lichen tissue. They might appear as white or colored deposits when you examine lichen sections under a microscope. These secondary compounds serve various functions: protection against UV radiation, defense against herbivores, antimicrobial activity. The crystals are part of the lichen's chemical arsenal, its biochemical adaptation to challenging environments. There's a group of lichens that looks and feels completely different from the structured, layered forms we've been discussing. Gelatinous lichens can appear jelly-like due to mucilaginous secretions. When wet, these lichens swell up, becoming soft and slippery, almost like seaweed or algae. The mucilage is produced by the photobiont, particularly when the photobiont is a cyanobacterium. This gelatinous matrix can hold enormous amounts of water, allowing the lichen to absorb and retain moisture very efficiently. When these lichens dry out, they collapse into thin, dark, almost unrecognizable crusts. But add water, and they spring back to life, swelling up again into their jelly-like form. Finally, consider the outer surface of many lichens. It can be water repellent, helping regulate moisture. This might seem paradoxical. Why would an organism that needs water have a water-repellent surface? The answer lies in moisture regulation. A completely water-absorbent surface would cause the lichen to become waterlogged during rain, potentially drowning the photobiont cells in too much water. A water-repellent surface allows the lichen to control moisture uptake, absorbing what it needs while shedding excess. It's about balance, about maintaining optimal moisture levels rather than simply maximizing water absorption. As you settle deeper into rest, picture the architecture of a lichen. The thallus, built mostly from woven fungal hyphae. Photobiont cells embedded within this structure, surrounded and protected. Layers when present, upper cortex filtering light and retaining moisture, photobiont layer capturing solar energy, medulla providing structure and storage, sometimes a lower cortex adding protection from below. No true roots or stems or leaves, just this elegant simplified structure. Attachment through rhizines or holdfasts. Sometimes crystals of chemical compounds scattered through the tissue. In some species, mucilage creating jelly-like texture when wet. A water-repellent surface helping balance moisture. Simple architecture supporting complex life. Let these images drift through your thoughts as sleep approaches. Lichens come in many forms, many shapes and growth patterns. Let's explore this diversity of forms you might encounter in the landscape. Crustose lichens form thin crusts tightly attached to rock or bark. These are perhaps the most common lichens in terms of species diversity, though they're easy to overlook because they're so flat and tightly bonded to their substrates. Imagine a rock surface covered with what looks like paint, patches of gray, orange, yellow, green, black. Look closer and you'll see these aren't paint but living organisms, crustose lichens spreading across the stone. They follow every contour of the rock surface, filling tiny pits, spreading across rough textures, creating a thin biological layer over the mineral substrate. These crustose lichens are so tightly attached that they cannot be removed without damage. Try to scrape one off a rock and you'll take rock with it, or you'll destroy the lichen in the process. This tight attachment is both strength and limitation. Strength because the lichen is firmly anchored, resistant to being dislodged by wind or rain or the passage of animals. Limitation because the lichen can never move, never relocate if conditions deteriorate. It's committed absolutely to the spot where its spores first germinated and found purchase. Foliose lichens have a completely different form. They have leaf-like lobes with distinct upper and lower surfaces. Foliose comes from the same root as foliage, it means leaf-like. These lichens spread across surfaces in overlapping lobes that can lift away from the substrate. They're not as tightly attached as crustose lichens. Instead, they're loosely attached at certain points, leaving the rest of the thallus free to expand and move slightly. Run your fingers across a foliose lichen on a tree trunk or rock and you can feel how some parts are attached while others lift away. This looser attachment gives foliose lichens more flexibility than crustose species. They can grow larger, spread farther, create more three-dimensional structures. Then there are fruticose lichens, which take the three-dimensional form even further. These are shrubby or hair-like and branch in 3 dimensions. Fruticose means shrubby, and that's exactly what these lichens look like, miniature shrubs or branching structures standing up from their substrate. Some fruticose lichens grow upright from rock or soil, creating small tufts or bushes. Others hang downward from tree branches. They're the most structurally complex lichens, with the most elaborate architecture. Some fruticose species, like Bryoria and Alectoria, hang in long beard-like strands. Walk through certain forests, particularly in regions with high humidity, and you'll see these hanging lichens draping from tree branches like pale green or dark brown beards. They can hang down for many inches, sometimes even feet, swaying gently in the breeze. These hanging lichens are often called old man's beard, though different species in different regions share this common name. They're particularly abundant in old-growth forests where tree branches have been stable for decades or centuries, giving the lichens time to grow to impressive lengths. Not all lichens fit neatly into the Crustosfolios fruticose classification though. Some have intermediate forms. Squamulose lichens have small scale-like lobes. They're like foliose lichens but on a much smaller scale, with tiny overlapping scales rather than large leaf-like lobes. They create textures that are neither completely crusty nor completely leafy, but somewhere in between. You might see them as small scaly patches on bark or rock, each scale attached at one point and lifted at the edges. There's another unusual growth form worth mentioning. Leprose lichens appear powdery and lack a defined cortex. These lichens don't have the layered structure we discussed earlier. They're not organized into neat cortex photobiont medulla arrangements. Instead, they're essentially loose associations of fungal hyphae and photobiont cells, creating a powdery or granular appearance. Touch a leprose lichen and you might get powder on your fingers. They seem almost unstructured, almost like the partnership is barely holding together. Yet they survive and reproduce, occupying their own ecological niches. Color adds another dimension to lichen diversity. While many lichens are gray or green, some stand out with vivid colors. Some fruticose species, such as Letharia vulpina, are bright yellow-green. This particular lichen, commonly called wolf lichen, grows on conifer branches in western North America and produces vivid chartreuse branches that seem to glow against the darker bark. The bright color comes from bulpinic acid, a chemical compound the lichen produces. Historically, this compound was used as a poison, particularly in Europe where it was added to meat baits to kill wolves and foxes, hence the name wolf lichen. Today we know it's toxic to mammals, but the lichen itself thrives, its bright color perhaps warning potential grazers away. One of the most striking things about many lichens is how they change with moisture. Many lichens dramatically change appearance when wet, becoming brighter and translucent. A lichen that looks dull gray or even black when dry might transform to bright green or vivid orange when rain soaks it. The transformation can be remarkably rapid. Within minutes of rain starting, dry-looking lichens come alive with color. This change happens because water fills the spaces in lichen tissue, allowing light to penetrate deeper, and because the photobiont cells become more visible when hydrated. The photosynthetic pigments, the chlorophyll and carotenoids, show their true colors when water is present. It's one of the delights of watching lichens after rain, seeing them shift from drab to brilliant as moisture brings them back to life. As you drift towards sleep, picture this diversity of forms. Crustose lichens painted across rocks, impossible to remove without damage. Foliose lichens spreading leafy lobes across tree trunks. Fruticose lichens branching in 3 dimensions, some hanging in long strands like beards from branches. Squamulose lichens with their tiny scales. Leprose lichens with their powdery, unstructured texture. Bright yellow-green Letharia vulpina glowing on conifer branches. And the transformations that happen with water, dull lichens becoming bright, dark lichens revealing hidden colors, all of them responding to moisture by changing their appearance. A landscape of forms, of colors, of textures. The diversity of lichen architecture, shaped by evolution and environment. Let these images settle into rest. Let's explore how lichens live with water, or more accurately, how they live with constantly changing water availability. Lichens are poikilohydric. That's a technical term describing organisms that have little control over their water content. Poikilo means varied or changing, and hydric refers to water. Poikilohydric organisms are those whose internal water content changes with environmental conditions rather than being regulated internally. Most animals and vascular plants are not poikilohydric. They maintain relatively constant internal water levels through various regulatory mechanisms. But lichens, lacking roots and cuticles and vascular systems, cannot regulate their water content. They simply equilibrate with their environment, becoming as wet or as dry as the conditions around them allow. They quickly equilibrate with the surrounding humidity. Put a dry lichen in humid air and it will absorb water from that air, swelling slightly, becoming more pliable. Put a wet lichen in dry air and it will lose water through evaporation, shrinking, becoming stiff and brittle. This equilibration happens rapidly, sometimes within minutes. The lichen doesn't choose to absorb or release water. It simply responds passively to the moisture gradient between itself and the air around it. It's completely at the mercy of environmental humidity. When dry, lichens enter a reversible state called cryptobiosis. This is one of their most remarkable adaptations. Cryptobiosis literally means hidden life. The lichen isn't dead when it's dry, but it's not actively living either. Metabolism stops. Photosynthesis ceases. Growth halts. The cells remain viable but dormant, waiting. It's like suspended animation, a pause in all life processes. The lichen can remain in this state for extended periods, days, weeks, months, even years in extreme cases. And it can survive conditions that would kill most organisms: extreme cold, high temperatures, intense radiation. In cryptobiosis, the lichen is protected, waiting for the return of water. Upon rehydration, photosynthesis can restart within minutes. This is the other side of the cryptobiosis strategy. The lichen doesn't need days or weeks to recover from desiccation. Add water and the metabolic machinery springs back to life almost immediately. Chlorophyll molecules begin capturing light again. Enzymes start catalyzing reactions. Carbon dioxide begins being fixed into sugars. The transformation from dormant to active can happen faster than you might think possible. It's as if the lichen was never dormant at all, as if it was just waiting for this moment to continue where it left off. This ability allows lichens to tolerate repeated cycles of drying and re-wetting. In many environments, this cycling is constant. Morning dew wets the lichen. Afternoon sun dries it out. Evening fog wets it again. Nighttime cooling dries it once more. Day after day, wet and dry, wet and dry. Most organisms couldn't survive this constant fluctuation, but lichens thrive on it. Each wet period is an opportunity for growth, however brief. Each dry period is just dormancy, survived without damage. Over time, through countless cycles, the lichen slowly accumulates biomass, slowly grows, slowly expands. Lichens absorb water and minerals directly from rain, fog, and dust. They have no roots reaching into soil to extract water and nutrients. Everything they need comes from above, from the air and from precipitation. When rain falls on a lichen, the water is absorbed directly through the surface. Dissolved minerals in the rainwater, calcium, magnesium, nitrogen compounds, phosphorus, are absorbed along with the water. Fog condenses on lichen surfaces and is similarly absorbed. Dust particles that settle on lichens can be dissolved by moisture and their mineral content absorbed. It's a completely different strategy from rooted plants, which draw water up from soil. Lichens are entirely dependent on what falls on them from the sky. But this dependency creates a vulnerability. Because they lack roots and cuticles, they are highly exposed to pollutants. A plant with roots might absorb some pollutants from soil or water, but it has barriers, cell membranes, selective uptake mechanisms, protective waxes on leaves. A lichen has none of these defenses. Whatever is in the rain or fog or airborne dust, the lichen absorbs it all. Sulfur dioxide from industrial pollution, heavy metals from mining or smelting, radionuclides from nuclear accidents, pesticides from agriculture. All of these can be absorbed by lichens, accumulated in their tissues, affecting their health and survival. This makes lichens excellent indicators of air quality, as we'll explore later, but it also makes them vulnerable to pollution. Given these constraints, the dependence on atmospheric moisture, the constant cycling between wet and dry, the slow pace of cryptobiotic life, it's not surprising that most lichens grow very slowly. Often less than 1 millimeter per year. 1 millimeter. That's about the thickness of a credit card. Imagine growing that slowly. A year of life, and you've added a layer the thickness of a credit card to your diameter. It's hard to comprehend growth that slow, time scales that extended. Many crustose species expand only 1 to 2 millimeters annually. These are among the slowest growing organisms on Earth. They're slower than trees, slower than most mosses, slower than almost anything else you might think of. This slow growth is partly a consequence of their environment. They often live in harsh conditions where growth is difficult. But it's also a consequence of their lifestyle. The constant cycling between active and dormant states means they're not always growing. They grow only when wet, which might be only a small fraction of total time. Growth happens in fits and starts, brief periods of activity between longer periods of dormancy. And here's something remarkable. Some individual lichens on rocks may be hundreds or even thousands of years old. Because they grow so slowly, and because they can survive in stable environments like rock surfaces where disturbance is rare, lichens can achieve great ages. There are documented cases of individual crustose lichens that have been dated to over 4,000 years old. 4,000 years. That's older than the pyramids of Egypt. Older than written history in most parts of the world. an organism that was already growing on a rock when ancient civilizations were just forming, and it's still there today, still slowly expanding, still surviving through the same patient strategy that has kept it alive for millennia. As sleep draws nearer, picture the patient life of a lichen. Constantly changing water content, equilibrating with the humidity around it. Entering cryptobiosis when dry, all life processes paused, waiting in suspended animation. Springing back to active photosynthesis within minutes when water returns. Cycling between wet and dry, dormant and active, hour by hour and day by day. Absorbing water and minerals directly from rain and fog and dust, with no protective barriers against whatever pollutants those sources might contain. Growing incredibly slowly, maybe 1 or 2 millimeters per year. And yet persisting, surviving, some individuals living for thousands of years on the same rock where their spores first landed. Patient beyond measure. Adapted to life at a pace we can barely comprehend. Rest now, and let the slow patience of lichens carry you into sleep. Let's explore how lichens reproduce, how these partnerships create new partnerships, how they spread to new surfaces, how they ensure their continuation across generations. The fungal partner in a lichen often reproduces sexually through specialized structures. In fruiting bodies called apothecia or perithecia. These are the reproductive structures of the fungus, the places where sexual reproduction occurs and spores are produced. Apothecia are typically cup-shaped or disc-shaped structures that appear on the lichen surface. They might be the same color as the rest of the lichen, or they might be different, bright red, orange, brown, black. Perithecia are flask-shaped structures embedded in the lichen tissue with just a tiny opening at the surface. Both structures serve the same basic function. They're where the fungus produces its sexual spores. These reproductive structures release spores into the air. Countless microscopic spores, each capable of starting a new fungal colony. The spores are dispersed by wind, carried on air currents to new locations. Some land on suitable substrates, rock, bark, soil. Some land in unsuitable places and die. It's a numbers game, a strategy of producing so many spores that some will inevitably find favorable conditions. The fungus is reproducing, scattering its genetic material across the landscape. But here's the challenge. After germination, a fungal spore must encounter a compatible photobiont. The spore germinates and begins growing fungal hyphae. But these hyphae alone cannot photosynthesize. They cannot make food. Without a photobiont partner, the fungus will starve. So the newly germinated fungal hyphae must, through pure chance, encounter the right kind of alga or cyanobacterium. One that's compatible, that can be incorporated into a partnership. The fungal hyphae must recognize the photobiont, surround it, establish the symbiotic relationship. Only then can a new lichen form. Only then can a partnership be recreated. This seems incredibly difficult, incredibly unlikely. How often does a fungal spore land near the right kind of algae? How many spores germinate only to die because they never find a partner? The odds seem stacked against success. And yet lichens are everywhere, suggesting that somehow, despite the odds, this process works often enough. But there's another strategy, one that doesn't depend on chance encounters between fungal spores and photobionts. Many lichens also reproduce vegetatively. Vegetative reproduction means creating new individuals without sex, without the fusion of gametes, without the production of sexual spores. It's cloning essentially, creating genetic copies of yourself through various mechanisms. Sordia are one of the most common structures for vegetative reproduction. These are tiny propagules of algal cells wrapped in fungal hyphae. Imagine a small package containing everything needed to start a new lichen, both the fungus and the photobiont already together, already in partnership. Sordia form inside the lichen thallus and are released through breaks in the cortex. They appear as a powdery coating on the lichen surface. Wind or rain can dislodge sordia and carry them to new locations. When a ceridium lands on a suitable substrate, it can simply start growing. No need to find a partner. The partnership already exists in miniature. Just growth and expansion needed. Isidia are another vegetative reproductive structure. These are small outgrowths that break off to form new lichens. They're like tiny versions of the parent lichen, projecting from the thallus surface. Eventually they break off, perhaps dislodged by wind or rain or the touch of an animal. Once detached, they can be transported to new locations. And like sordia, isidia already contain both partners. They're pre-packaged lichens, ready to establish themselves wherever they land. These vegetative reproductive strategies, sordia and isidia, solve the problem of getting fungus and photobiont together. No need for chance encounters. The partners stay together through the reproductive process, ensuring that new lichens have everything they need from the start. But not all lichens use vegetative reproduction. Some species rely mainly on fungal spores. These species are betting on the sexual reproduction strategy, despite its challenges. They produce abundant spores, release them into the air, and trust that some will find compatible photobionts. It's a riskier strategy in some ways, but it has advantages. Sexual reproduction creates genetic variation through recombination of genes from two parents. This variation provides raw material for evolution, helping the species adapt to changing conditions. Vegetative reproduction creates genetic clones, which is efficient but doesn't generate new variation. Certain crustose lichens have evolved an interesting variation on sexual reproduction. They're self-fertile, a condition called homothallic. In most fungi that reproduce sexually, you need two compatible mating types to come together to produce sexual spores. It's similar to having male and female in animals, though fungal mating systems are more complex. But homothallic fungi don't need two mating types. A single individual can complete the sexual cycle on its own, producing spores without needing to encounter another compatible individual. This might seem like it defeats the purpose of sexual reproduction. If you're just fertilizing yourself, where's the genetic variation? But it does allow for spore production even in isolation, even when no other compatible individuals are nearby. Self-fertility may help survival in isolated or harsh environments. Imagine a lichen spore that lands on an isolated rock outcrop, far from any other lichens. If it's heterothallic, requiring a partner for sexual reproduction, it might never be able to produce sexual spores, because no compatible partner ever arrives. But if it's homothallic, it can produce spores on its own. This allows it to disperse its genes despite being isolated. It's an adaptation to the reality that lichens often colonize isolated locations where finding a sexual partner would be impossible. Finally, there's an often overlooked mechanism of dispersal. Lichen fragments can travel long distances by wind, water, animals, or humans. A piece of lichen broken off by a storm can be blown for miles. A fragment caught on an animal's fur can be transported to a new location. Humans moving through lichen-rich environments can carry fragments on their clothing or equipment. These fragments, if they contain both fungal and photobiont cells, can potentially establish new lichens wherever they land. It's not exactly reproduction, more like accidental propagation. But it's effective, helping lichens colonize new areas, maintaining genetic connections between distant populations. As you settle into deeper rest, picture these reproductive strategies. Apothecia and parathecia releasing countless spores into the air, each spore carrying only the fungal genes, hoping to encounter a compatible photobiont after germination. Sorgia forming inside the lichen, tiny packages of both partners ready to be dispersed together. Isidia projecting from the surface, breaking off to form new lichens. Homothallic species producing sexual spores without needing a partner. Fragments traveling on wind and water and animals, accidentally spreading the partnership to new locations. Multiple strategies, multiple mechanisms, all ensuring that these partnerships continue, that lichens persist across landscapes and through time. Let these images of reproduction in stillness drift through your thoughts as sleep approaches. Let's explore where lichens live and the remarkable roles they play in some of Earth's harshest environments. Lichens colonize extreme environments with high light, cold, heat, or dryness. Places where most other organisms struggle or fail entirely. Arctic tundra where temperatures plunge far below freezing and wind scours the landscape. Hot deserts where surface temperatures can exceed 60 degrees Celsius and rain might not fall for months or years. High mountain peaks where intense solar radiation, cold temperatures, and thin air create brutal conditions. Antarctic rocks where only the hardiest organisms can survive. In all these extreme environments, lichens are often among the few visible life forms, the pioneers that can tolerate conditions that exclude almost everything else. They grow on an amazing variety of substrates. Rock, of course, lichens are famous for covering rocks in colorful crusts. But also soil, bark, leaves, wood, bone, concrete, and even metal. If a surface is relatively stable and receives some light, lichens can potentially colonize it. They are not particular about chemistry or texture. Granite or limestone, acidic or alkaline, smooth or rough, lichens find ways to attach and grow. Concrete buildings in cities host lichens. Metal fences and railings can support lichen growth. Animal bones left on the landscape can be colonized. Even living leaves in tropical rainforests sometimes support specialized leaf-dwelling lichens. The diversity of substrates lichens can use is remarkable. Many lichens are pioneer species on bare rock. After glaciers retreat, leaving behind raw scraped bedrock. After volcanic eruptions deposit fresh lava that cools into bare stone. After quarrying or construction exposes new rock surfaces. In all these situations, lichens are often the first complex organisms to arrive. Their spores or vegetative propagules land on the bare rock and begin growing. They don't need soil. They don't need organic matter. just rock and air and occasional moisture. They're the colonizers, the first wave of life returning to lifeless surfaces. And once established, lichens contribute to an important geological process. Lichen acids slowly dissolve minerals, contributing to weathering. The lichen produces organic acids, various lichen compounds plus carbonic acid from respiration. These acids contact the rock surface, slowly dissolving minerals, etching into the stone. The process is extremely slow, measured in millimeters per 1,000 years. But it's real. Lichens are literally eating the rock, breaking down minerals to extract nutrients like calcium, magnesium, iron. And in doing so, they begin the long process of converting rock to soil. They create small pits and roughened surfaces where dust can accumulate. They contribute organic matter when they die. They start the soil formation process that will eventually allow other organisms to colonize. In tundra and alpine regions, lichens form significant ground cover. Walk across the Arctic tundra and your boots crunch on lichens, reindeer lichens, Cetraria species, various crustose forms coating rocks. Climb above treeline in mountains and again, lichens are everywhere. They cover rocks, grow on soil, create a significant fraction of the visible biomass. In these harsh environments where growing season is short and conditions are difficult, lichens' ability to tolerate cold, to grow slowly but persistently, to survive long dormant periods, gives them advantages over many other organisms. They dominate through patience and tolerance. In forests, particularly humid forests, epiphytic lichens create intricate communities on branches. Epiphytic means growing on plants without parasitizing them. Tree branches in old-growth forests can be covered with lichens, foliose species spreading across bark, fruticose species hanging down, crustose species filling in the gaps. These epiphytic communities can be remarkably diverse, with dozens of lichen species on a single old tree. They use the tree as a platform, a place to access light, but they take nothing from the tree. They're just using the bark as substrate, like they might use a rock. In deserts, lichens play a different but equally important role. Desert soil crust lichens stabilize soil and affect water infiltration. In many deserts, the soil surface is covered with biological soil crust, a living layer composed of lichens, mosses, cyanobacteria, and other organisms all bound together. This crust stabilizes the soil, preventing wind erosion. Without the crust, desert soils would blow away, creating dust storms and degrading the landscape. The crust also affects how water behaves. It can either enhance or impede water infiltration depending on the specific crust composition. It's a crucial component of desert ecosystems, and lichens are often major contributors to these crusts. Some lichens bring an additional gift to nutrient-poor environments. Cyanobacterial lichens enrich nutrient-poor habitats by fixing nitrogen. Remember that some lichens partner with cyanobacteria, and cyanobacteria can fix atmospheric nitrogen. In environments where nitrogen is limiting, bare rock, sandy soil, alpine areas, this nitrogen fixation can be crucial. The lichen grows, fixes nitrogen, and when it dies or sheds portions of itself, that nitrogen becomes available to other organisms. The lichen is enriching the environment, making it possible for other life to follow. Lichens themselves host other organisms. They host microscopic invertebrates and fungi. Tiny mites live in lichen thalli, grazing on the photobiont cells or on fungi that grow on the lichen. Tardigrades, water bears, can be found in lichens, surviving desiccation along with the lichen and becoming active when moisture returns. Tiny insects live in the cracks and crevices of fruticose lichens. Fungi other than the lichen mycobiont can colonize lichen thalli, sometimes parasitizing the lichen, sometimes just living alongside it. The lichen is habitat, a three-dimensional structure that provides shelter and food for organisms too small to see without magnification. And some larger animals depend on lichens too. Reindeer and caribou rely on certain lichens as winter forage. In the deep winter when snow covers other food sources, reindeer dig through snow to reach lichen-covered ground. They eat the lichens, particularly species of Cladonia commonly called reindeer lichens. These lichens provide calories and some nutrients during the harshest months. The reindeer have even evolved digestive systems that can break down lichen compounds that other herbivores cannot digest. It's a crucial relationship in Arctic ecosystems, one that has sustained reindeer populations for thousands of years. As you drift towards sleep, picture lichens in their many roles. Colonizing bare rock in the wake of glaciers and volcanoes. Producing acids that slowly dissolve minerals and begin soil formation. Covering the ground in tundra and alpine regions, creating significant biomass in harsh environments. Draping from forest branches in intricate epiphytic communities. Stabilizing desert soils in biological crusts. Fixing nitrogen in nutrient-poor habitats. Hosting microscopic invertebrates and fungi. Feeding reindeer in winter. Pioneers and engineers, food and habitat, surviving and thriving in extreme worlds where few other organisms can persist. Let these images of lichens as pioneers settle into your thoughts. Rest now in the knowledge that life finds ways, even in the harshest places, and lichens are often leading the way. Let's explore how lichens serve as indicators and monitors of environmental conditions, particularly air quality. Lichens are widely used as bioindicators of air quality. A bioindicator is an organism whose presence, absence, abundance, or health tells you something about environmental conditions. And lichens are among the best bioindicators we have for air pollution. They're sensitive, they're visible, they accumulate pollutants in measurable ways, and they're present in most environments, making them excellent tools for monitoring air quality across large areas. Many lichen species are sensitive to sulfur dioxide and other pollutants. Sulfur dioxide was historically the most important air pollutant affecting lichens, produced primarily by burning fossil fuels, particularly coal. When sulfur dioxide dissolves in moisture on lichen surfaces, it forms sulfurous acid, which damages cells, inhibits photosynthesis, and eventually kills the lichen. Different species have different sensitivities. Some can tolerate relatively high sulfur dioxide levels. Others decline when pollution reaches even moderate levels. The most sensitive species disappear first as pollution increases. Polluted urban areas often show reduced lichen diversity. In cities with significant air pollution, particularly during the industrial era when coal burning was common, lichen diversity would drop dramatically. Surveys in polluted cities might find only one or two hardy species, or no lichens at all in the most polluted areas. The forests and rocks that should host dozens of lichen species would be barren, covered only with bare bark or stone. This lichen desert phenomenon became well documented in many industrial cities during the 20th century. But here's something interesting. Some tolerant species may persist or increase in polluted areas. While sensitive species disappear, hardy species that can tolerate pollution might actually become more abundant, facing less competition from the species that have been eliminated. So you might see strange patterns in polluted areas, very low total diversity, but very high abundance of a few tolerant species. These tolerant species serve as indicators too. Their presence in abundance suggests pollution levels high enough to exclude sensitive species. Lichens don't just respond to pollution by being present or absent. They accumulate heavy metals and radionuclides from the atmosphere. Lichens have no mechanisms to exclude or detoxify pollutants. They absorb everything that's in the air and rain. Heavy metals like lead, copper, zinc, cadmium. Radionuclides like cesium-137 or strontium-90. All of these accumulate in lichen tissue. The lichen becomes a living record of what's in the environment. Scientists can collect lichens, analyze their metal content, and use this data to map pollution levels across landscapes. This characteristic makes lichens valuable tools. Scientists use them to track environmental contamination. After the Chernobyl nuclear accident, for instance, lichens across Europe absorbed cesium-137 from the radioactive fallout. By analyzing lichens, scientists could map the spread of contamination, identify hotspots, track how radioactivity moved through ecosystems. Similar studies have been done around other pollution sources, mines, smelters, industrial facilities. The lichens tell the story of what they've been exposed to. Lichen studies can also document historical pollution trends. Because lichens are long-lived and accumulate pollutants over their lifetime, analyzing lichens of different ages or from different time periods can reveal how pollution has changed. In regions where air quality has improved, where coal burning has decreased or where pollution controls have been implemented, lichen diversity has recovered. Species that were absent for decades have returned. Areas that were lichen deserts have become lichen-rich again. Long-term lichen monitoring provides evidence of these environmental changes. But lichens don't just respond to and indicate pollution. They also influence their environment in various ways. Lichen cover influences surface temperature and moisture. A rock or tree branch covered with lichen has different thermal properties than a bare surface. The lichen insulates, moderating temperature fluctuations. It holds moisture, creating a more humid microenvironment at the surface. These effects might seem small, but they can influence what other organisms can colonize the surface, how quickly weathering proceeds, how the microclimate develops. Lichens also play a role in carbon cycling. They sequester carbon in their biomass. The photobiont captures carbon dioxide from the atmosphere and fixes it into sugars. Some of this carbon is incorporated into lichen structure. When lichens die, this carbon enters the soil or the ecosystem where the lichen grew. In ecosystems where lichens are abundant, tundra, some forests, alpine areas, the amount of carbon sequestered in lichen biomass can be significant. Not as much as in trees, of course, but measurable and meaningful. Finally, in many habitats, lichens help maintain biodiversity. They provide habitat for invertebrates, food for certain animals, substrate for other organisms. They contribute to the structural complexity of ecosystems, creating three-dimensional surfaces, modifying microhabitats. Remove lichens from an ecosystem and you'd lose not just the lichens themselves but also the organisms that depend on them. In this way, lichens are keystone organisms in some ecosystems. Their presence supports other species, maintains ecosystem function, contributes to overall diversity. As sleep deepens, picture lichens as environmental witnesses. Sensitive species disappearing as pollution increases, creating lichen deserts in polluted cities. Tolerant species persisting or increasing in abundance. Lichens accumulating heavy metals and radionuclides, becoming living records of contamination. Scientists collecting them to map pollution patterns. Long-term studies documenting how lichen communities recover as air quality improves. Lichen cover moderating surface temperature and moisture. Carbon being sequestered in lichen biomass. Biodiversity being maintained by lichen presence and the habitat complexity they create. Silent witnesses to environmental conditions, indicators of change, contributors to ecosystem function. Let these images drift gently through your thoughts. Rest now, knowing that organisms all around us are recording environmental conditions, telling stories about air and water and landscape, if we just know how to read them. Let's explore the remarkable chemistry of lichens, the unusual compounds they produce, and what these substances might offer to human medicine. Lichens produce a remarkable array of unique secondary compounds. Secondary compounds are chemicals that organisms produce that aren't directly involved in basic metabolism, not part of photosynthesis or respiration or basic cell structure. Instead, they serve specialized functions: defense, protection, communication. Lichens are champions of secondary compound production. They produce hundreds of different substances that are found nowhere else in nature. These lichen substances have unusual chemical structures, unusual properties, and as we're discovering, unusual biological activities. Many of these compounds are stored as crystals in the outer layers of the lichen. If you examine a lichen section under a microscope, you might see these crystals scattered through the tissue, white or colored deposits accumulated between cells or in the cortex. The lichen produces these substances and then precipitates them out as crystals rather than keeping them dissolved. This storage strategy might protect the lichen cells from the potentially toxic effects of high concentrations of these compounds. It might also make the compounds more available for their defensive functions, creating a chemical barrier right at the lichen's surface. Here's something remarkable that scientists have discovered through testing lichen compounds. About half of lichen species possess antibiotic properties. Half. That's an extraordinary proportion. Test extracts from randomly selected lichens against bacteria, and about half will show some ability to inhibit bacterial growth. This suggests that antibiotic activity is widespread in lichens, not confined to a few unusual species but common across the group. Many of these antibiotic compounds are especially active against gram-positive bacteria. Gram-positive bacteria include many human pathogens. Staphylococcus species that cause skin infections, Streptococcus species that cause strep throat and other infections, Bacillus species, Clostridium species. The fact that lichen compounds are active against these bacteria raises the possibility that they could be useful in human medicine, either as antibiotics themselves or as templates for developing synthetic antibiotics. Some lichen compounds show antioxidant and anti-inflammatory effects. Antioxidants are substances that can neutralize harmful reactive molecules called free radicals, potentially protecting cells from damage. Anti-inflammatory compounds reduce inflammation, the body's response to injury or infection. Both properties have potential medical applications. Chronic inflammation is implicated in many diseases. Oxidative stress contributes to aging and various pathologies. Compounds that can address these issues are valuable. Even more intriguingly, certain lichen extracts have demonstrated anti-tumor activity in laboratory studies. When tested against cancer cells in culture, some lichen compounds can inhibit cell growth, induce cell death, or interfere with the processes that allow cancer cells to proliferate. This doesn't mean lichens are a cure for cancer. Laboratory studies are just a first step, and many compounds active in lab dishes don't work in living organisms. But it does suggest that lichen chemistry might offer leads for developing cancer treatments, molecules worth investigating further. One specific lichen compound has already found medical use. Usnic acid has been used in topical antibiotic preparations. Usnic acid is a yellow compound found in many lichens, particularly those in the genus Usnea. It has strong antibiotic activity, particularly against gram-positive bacteria. In some countries, usnic acid-containing ointments and creams have been developed for treating minor skin infections and wounds. It's one of the few lichen compounds that has made the transition from traditional use to modern pharmaceutical application. Another lichen compound with interesting properties is the yellow pigment parietin. This substance protects against UV radiation. Lichens that grow in highlight environments, on exposed rocks, in alpine areas, in deserts, often produce parietin and related pigments. These yellow, orange, or red compounds absorb UV light, preventing it from reaching and damaging the photobiont cells beneath. It's the lichen's own sunscreen, chemically protecting the sensitive photosynthetic cells from UV damage. And humans are now exploring whether this natural sunscreen could work for us too. Parietin is being explored as a potential natural sunscreen ingredient. Could we extract parietin from lichens, or synthesize it, and incorporate it into sunscreen formulations? It's a natural UV-absorbing compound, potentially less controversial than some synthetic sunscreen chemicals. Research is ongoing, testing whether parietin is safe and effective when applied to human skin, whether it provides adequate UV protection, whether it can be produced sustainably. There's a practical reason lichenologists care about lichen chemistry beyond its medical potential. Lichen chemistry is often species-specific and aids identification. Many lichen species that look identical can be distinguished by their chemistry. Two lichens might have the same growth form, the same color, the same microscopic structures. But if you test them for specific compounds, you find one produces usnic acid while the other produces a different substance. This chemical difference might be the only reliable way to tell them apart. Lichenologists use chemical spot tests, applying reagents to lichen tissue and observing color changes, to help identify specimens. They use thin layer chromatography to separate and identify compounds. Chemistry becomes an identification tool, as important as morphology. As you settle into deeper rest, picture this hidden chemistry. Hundreds of unique compounds produced by lichens. Crystals of these substances stored in lichen tissues. About half of all lichen species with antibiotic properties. Compounds active against bacteria, showing antioxidant effects, demonstrating anti-inflammatory activity, inhibiting tumor cells in laboratory tests. Usnic acid already in medical use in topical preparations. Parietin protecting lichens from UV radiation and potentially offering similar protection to humans. Chemistry being used to identify species that look identical. A vast library of natural compounds, many still unexplored, some potentially valuable for medicine. The hidden chemistry of lichens, evolved for their own survival, potentially offering benefits to ours. Let these images drift through your mind as sleep approaches. The molecular machinery of lichens, producing substances found nowhere else, protecting themselves through chemistry, and perhaps offering us new tools for healing. Let's conclude our journey by exploring how humans have interacted with lichens throughout history and across cultures. Many cultures have used lichens medicinally. Long before modern chemistry could identify and isolate lichen compounds, people were using whole lichens or lichen preparations to treat various ailments. Traditional medicine systems around the world incorporated lichens, recognizing through experience that these organisms had healing properties. Historical records and ethnobotanical studies document that lichens have treated ailments such as tuberculosis, ulcers, and skin problems. In Europe, Iceland moss, actually a lichen, not a moss, was used for treating tuberculosis and other respiratory complaints. Various lichens were applied to wounds and skin ulcers, either as poultices or in ointments. The antibiotic properties we now know many lichens possess likely contributed to these effects, helping wounds heal, preventing infection. Some traditional medicine systems used lichens as stomachic, anti-diabetic, or hemostatic remedies. Stomachic remedies are those that aid digestion or soothe the stomach. Certain lichens were consumed as teas or powders for digestive complaints. Anti-diabetic use suggests lichens being employed to manage blood sugar or symptoms of diabetes, though whether they were actually effective is unclear. Hemostatic remedies are those that stop bleeding. Lichen preparations were sometimes applied to wounds specifically to staunch blood flow, and some lichens do contain compounds that might aid clotting. But humans haven't just used lichens as medicine. Some have also eaten them. Certain species were eaten as emergency food, such as bread moss in northern Europe. Iceland moss again appears in historical accounts, this time as a famine food. During times of scarcity, when crops failed or other food sources were exhausted, people would collect lichens, process them to remove bitter compounds, and cook them into gruel or bread. The lichens provided some calories and nutrients, enough to help people survive until better food became available. It was hardly a preferred food, the processing was laborious, the taste often poor, the nutritional value modest. But it was food when food was desperately needed. In other parts of the world, lichen consumption wasn't just emergency food but part of regular diet. Indigenous peoples consumed prepared bryoria and other lichens. Bryoria species, those long hair-like lichens hanging from trees, were harvested by various Native American groups in western North America. The lichens were collected, processed by steaming or pit cooking to remove acids and improve digestibility, and then consumed. They provided carbohydrates and some other nutrients. The preparation was careful and specific. Raw bryoria contains acids that would make you sick, but proper processing makes it edible. This knowledge, passed down through generations, allowed people to utilize a resource that might otherwise seem completely inedible. Lichens have also been used in food preparation more indirectly. They have flavored beers and fermented beverages. In some brewing traditions, lichens were added during fermentation to impart particular flavors or to clarify the brew. The compounds in lichens could affect fermentation processes, could contribute bitterness or other flavor notes, could help precipitate unwanted particles out of the liquid. It's a small footnote in brewing history, but it shows that humans have found many uses for these organisms. Another important historical use was in dye production. Lichens have produced natural dyes in yellows, oranges, purples, and browns. Certain lichens, when processed appropriately, yield vibrant dyes that can color wool, silk, and other fibers. The purple dyes were particularly valuable historically. Before synthetic dyes, getting a good purple color was difficult, and lichen-derived purples were among the best available. The famous Harris tweed from Scotland has traditionally used lichen dyes for some of its colors. The process of extracting dyes from lichens could be complex, sometimes requiring fermentation or treatment with ammonia, but the results were valuable enough to make the effort worthwhile. One specific dye derived from lichens had broader scientific importance. Some species provided litmus dye for pH testing. Litmus, that familiar substance that turns red in acid and blue in base, originally came from lichens. The scientific use of litmus as a pH indicator depended on this lichen-derived substance. Even today, though litmus can be synthesized, its name recalls its lichen origins. More recently, scientists have discovered potential medical applications for lichen compounds. At least one lichen compound has inhibited HIV growth in laboratory tests. HIV, the virus that causes AIDS, has proven difficult to treat with single drugs. It evolves rapidly, developing resistance. Any new compounds that show anti-HIV activity are worth investigating as potential components of combination therapies. The fact that a lichen compound can inhibit HIV in laboratory conditions doesn't mean it's ready to be a drug. There's a long path from laboratory activity to approved medicine. But it's a start, a lead worth following. Finally, lichens appear in art and folklore, demonstrating that humans have noticed and appreciated these organisms beyond their practical uses. Ernst Haeckel's detailed 1904 illustrations are perhaps the most famous artistic representations of lichens. Haeckel was a German biologist and artist who created incredibly detailed illustrations of various organisms. His lichen illustrations, part of his larger work Art Forms in Nature, show lichens with almost surreal beauty and precision. They were meant to be scientific illustrations, accurately depicting species and structures. But they're also works of art, compositions that highlight the geometric patterns, the intricate structures, the strange beauty of lichens. These illustrations helped bring lichens to broader attention, showing that these easily overlooked organisms were worthy of aesthetic appreciation. In folklore, lichens appear less prominently than plants or animals, but they're not absent. The name Old Man's Beard for hanging lichens connects them to human imagination. Some traditions attributed medicinal or magical properties to certain lichens. The slow growth and great age of some lichens has occasionally inspired stories or metaphors about time and patience. Lichens haven't captured human imagination the way flowers or trees have, but they've been noticed, named, woven into human culture in small ways. As you drift into final deep sleep, let these human connections settle into your thoughts. Lichens used for millennia as medicine, treating respiratory ailments and wounds and various other complaints. Eaten as emergency food in Europe, regularly consumed by some indigenous peoples in North America. Used to flavor beers, to dye fabrics in vibrant purples and yellows. Providing litmus for scientific pH testing. Showing activity against HIV in laboratory tests. Illustrated beautifully by Ernst Haeckel. Appearing in names and folklore. Organisms that exist independently of humans, but that humans have nonetheless found countless ways to use and appreciate. The human story and the lichen story intersecting in medicine, food, dyes, art, and culture. Rest now, and let the quiet partnership of lichens, with their photobiont, with ecosystems, with humans, carry you gently into sleep. The partnership that has existed for hundreds of millions of years, that covers rocks and trees across the planet, that survives in the harshest environments, that produces remarkable chemistry, that has fed and healed and colored human experience. Rest in the knowledge of this partnership, this cooperation, this successful collaboration that makes lichens possible and gives them their unique place in the living world.